His little Ohio, and he went to Brown University for his bachelor's, and then he proceeded to get not only a master's of philosophy, but also a master's of art at Columbia, while he also got his PhD there, working with Joel Craycraft and Shaim Naim. After that, he went and did a postdoc with Ben Winger, one of our former bird students here in the museum at the University of Michigan. And at that time, a position opened up in the Department of Environment and Sustainability, and he became a faculty member in 2019. Got that right? Yeah. So his work covers a lot of different things. One of the things he's done, he's worked with people like former students of ours, like Shane Dubé, on various projects. He's also very well known for being one of the driving forces in working with Dave Willard and all the bird folks associated with the window kill data analyses that have been done and published. And so it's a real pleasure to have him here today. And we're looking forward to hearing him talk about AI and metabolism and climate in birds.
Yeah, thanks, John. I'm extremely excited to be here. As John mentioned, for the last 7 or so years, a lot of my research focus has been based on questions that we've used the Field Museum salvage specimen to address. So I've been working with Field Museum specimens for a really long time, and I've never been to the Field Museum, which always seemed a little wrong. So I'm thrilled to be here. And I'm going to talk today about how I've been linking AI and specimens to try to address some questions about the linkages between climate and morphology in birds. But I wanted to start. I wanted to start by providing a little bit of context, which will be deeply intertwined with the story of the Field Museum specimens. So this really, my interest in this, what I'm going to talk about today, really started with Field Museum specimens about 7 years ago, and thinking about how species have responded to climate change. So really like this figure from a recent paper showing 2 of the major axes of adaptation to climate change that I think we all think about pretty regularly. So on the left and in the middle, you have shifts in range. So each of these colored bars is the trajectory of a species through time. And so you can see that species have shifted their ranges poleward and upward in elevation. And then all the way to the right, you have changes in phenology. See that species have shifted the timing of major life history events, so things like migration and the onset of breeding. And I think we are all probably pretty familiar with this idea as ways that birds respond to climate change and other global changes. So I think of these as niche tracking strategies, but we're also sort of interested in how a third axis of adaptation has manifested itself potentially within birds of North America. So can we think about how species have also changed their morphology and kind of changed in place in response to shifting environmental conditions? And so we really set off to work on this a long time ago with lots of North American birds, using the salvage specimen series here at the Field Museum and using a subset of that specimen series that included just over 70,000 birds from 52 species. So these are the well-sampled birds within the salvage specimen series, and looking at how they've changed over the past 40 years. All of this, of course, is based on morphological trait data that Dave measured by himself over 40 years with a whole bunch of other people, as I understand it, supporting that effort. But essentially what we have is a long-term, I think globally unique, long-term specimen series that we can use to look at morphological change through time. So we started looking at the specimens, and some really interesting things popped out. So here you see data from the 9 most well-represented species in that data set. And on the x-axis is time, on the y-axis is an index of size. And you can see that these species have all gotten smaller through time. This is a really consistent trend across all 52 species. And we looked at this with some multivariate model approaches and identified temperature on the breeding grounds as a potentially important driver of these changes. Of course, it was correlative. But thanks to the density and the precision of the data, we weren't restricted to just looking at long-term trends in morphology for these groups. We actually zoom in and characterize short-term fluctuations in body size through time. And when we looked at the short-term fluctuations in body size through time and compared them to short-term fluctuations in temperature, we found this really interesting tight negative correlation within these short-term fluctuations between the temperature and size data. So as temperatures briefly went down, body size would go up. And then the year after temperatures started to go up again, body size would start to go down. So of course, we're a bit constrained in thinking about causality with observational data, but it seemed like pretty good evidence that something related to shifting temperatures was driving changes in size in birds. We found other things like wing lengths have increased really broadly across these species. And we had no idea why that was happening. We couldn't explain it. And this totally sent us down a crazy rabbit hole. It's lasted many, many years and is ongoing. And we've found things like birds are getting smaller and longer winged. Phenology has shifted, but that doesn't seem to explain changes in wing length. Smaller species are changing faster than bigger species. Drivers of wing length change really are hard to understand, so we failed again trying to explain them. And there does seem to be a genomic basis for the shifts in some of these traits, in particular bill length, but not in traits like body size and wing length. So we're still grappling with all of these questions that are coming out of this data. And I just wanted to highlight that everything that I'm showing here and talking about here is work that I've done that wouldn't be possible without Dave's work. And it's all been done with Ben Winger. And we've had a great postdoc, Marketa Zamova, who led quite a bit of it. So this is all just to say, I've been really interested in the relationships between climate and morphology for a long time, largely as a result of what we've found in this long-term specimen series. And that has inspired me to do what I'm going to talk about today. And this is all new stuff. And so I've never talked about it before, and I'm curious to see how it goes. Hopefully it'll go well. But generally speaking, there are three parts to this story that I want to tell today. And they're all about trying to understand climate and morphology links more broadly in birds. So first, I'm going to talk about building a skeletal trait data set with Skelevision. Then I'm going to talk about the analysis of the relationship between climate variability and body plan evolution in passerine birds using the Skelevision data. And then I want to end by showcasing a couple of ways that this new type of data at a really big scale has let us, I think, develop a bit more of a mechanistic understanding of some long-standing observations about patterns in the natural world. So first, I want to talk about Skelevision, and I can't do that without talking about David Foey. So in 2019, just before COVID, I walked to the north part of our campus and met with David, who his bread and butter at the moment is trying to teach robots what can and cannot be opened within a room. So like, can you open a drawer or a door? Robots have a hard time with that, apparently. So that's what he works on. He's a true computer scientist, and he works with computer vision. And I dumped out a box of bones on his desk, and his eyes lit up, got super excited, and he really engaged with this project and has been engaged with it since 2019. And so nothing that I'm talking about today would have been possible at all without his contributions. And the reason that Skelevision came about is because when I was a PhD student at the American Museum of Natural History, I spent a lot of time measuring functional traits on skin specimens in the collection. And I did that for my dissertation. I also did that to contribute to a long-term effort led by Joe Tobias to measure this set of standardized traits on every species of bird in the world. And so here in this figure, you see a plot of the major functional trait data sets through time. That blue line is showing you a sort of a zero line, and everything above that is reflecting the number of species in a data set. Everything below it is reflecting the number of specimens. So you have two dots connected by a line for each data set. And the size of the dot is scaled to represent the number of traits in that data set. So the takeaway here is that through time as a community, we have been measuring traits on mostly skin specimens throughout the last century. We've been getting bigger and bigger trait data sets. And then AvoNet came in. These are actually are both AvoNet-based studies. And it really sort of changed the game. So now we have this comprehensive trait data set for all birds, which is really cool. It's been used a lot. And it was only possible because of a Herculean effort by Joe Tobias, but also over 100 people contributed to developing this data set, and it took more than a decade. So this was a major long-term investment in measuring these external traits to understand external morphology of birds. You can compare that to skeleton-based data sets. This is the same style figure where the two dots are one data set. the distance of the top dot above the line is the number of species, the distance below is the number of specimens. And you can see that the growth in skeletal trait data sets has been rather anemic. And I think this is attributable to the fact that skeletons are really annoying to work with. So in sum, if I sit back and look at my experience with these two types of data, I sort of think that one realization that I've had is that measuring traits by hand in skins and with skin collections has been really inefficient. So I would get a list of 1,000 species that are missing from AvoNet, and I would go into the collection and try to find those species and then measure them and then put them back. And the vast majority of my time was spent finding the specimens and putting them back, and a little bit of time was spent measuring them. So that was true with the skin measurements. And with skeletons, that's even more of a problem. So if you've ever dumped a skeletal specimen, a totally disarticulated skeletal specimen out, measured a few traits, and then put it back, you know what I'm talking about. A lot of time is spent very carefully moving little bones back into a box. So it's an even bigger problem with skeletal specimens. But this seemed like a problem that could be solved with computer vision. And this was kind of like a COVID fever dream project to begin with. So this is the original image capture setup that was built in my garage in the middle of COVID. And it's a pretty straightforward apparatus. So it has a flexible surface. You can take your skeletal specimen, dump it onto the surface, photograph it with this camera that's fixed above the surface, and then you can push the middle of the front of the surface and it forms this chute. You can brush the bones back into the box. So a process that might have taken 10 minutes or more now takes about a minute and a half. So in about a minute and a half, we can go from start to finish photographing a specimen. So now, instead of trying to find individual specimens, we went to the University of Michigan Museum of Zoology, and we started at one end of the pasturines and just did the whole collection. And so we weren't wasting any time looking for things, and we really reduced the amount of time it took to get things out and work with them. But what you're left with is not that useful. So this is an average image of a UMMZ skeletal specimen. The bones are spread haphazardly, so they're never in the same place across images. But we can train computers to measure these elements for us. So we've trained models to identify which pixels in this image are and are not bone. We've trained models to outline the different elements, so basically trace the outline of the different elements. And then we've trained a model to identify the different elements. So you can go from this picture to an identified and segmented out image of all of the elements of the skeleton without doing anything, without a human doing anything. And then we can measure things like the length of the elements. And that's what I'm going to talk about today. So we had someone do this at the UMMZ Museum, and it was one person. He worked for 4 months on it, and this was the result. So we were able to generate a really big comparative skeletal trait data set in a really short period of time. We have since expanded, and we've done some work in the Field Museum. We've photographed another 2,000-ish specimens. Hopefully, we'll expand that soon. So now the data set includes 14,000 specimens from about 2,000 species of passerines. So here you see a tree of all passerines, and the blue bars on the outside are species for which we have photographed at least one specimen. The height of the bars is scaled to reflect the number of specimens that we photographed, and it's a log scale. So it's really uneven sampling because, again, we just started at one end of the collection and did everything. So some of these species, we've got one individual that we measured. Some of them, we've got 100. And on each specimen, those are the bones. So the skeleton figure there, you can see there's some dark bones. Those are the bones that we've measured. So we've measured 12 elements from across the skeleton that give us an understanding of the gross morphology of these birds. I'm probably going to lapse into calling it the body plan, which is not the most precise term. But when I say the body plan of a bird, I basically mean the accumulation of information from all of these different elements of the skeleton. Then you see a map of our sampling over there with the colors reflecting the richness, the species richness within the data set. So if you're familiar with bird diversity, this is not such a terrible subsampling of bird diversity in the Americas. There are some notable gaps, like New Guinea, for example, where we have almost no diversity represented collection. So it's a global data set, but it's biased in important ways. Okay, so I wanted to introduce this because it's the basis for all the interesting stuff that I'm going to talk about. But I also learned a couple of things that I thought might be interesting to museum-based researchers throughout this process. The first that I think is really exciting is collecting data and generating data with ScalaVision is fast. So it took us about 1.5 or 2 minutes per specimen to measure 12 traits. But that's nothing compared to what you could do with the existing image data set that we have now. So even as the database of images grows, the amount of information that a human needs to provide a model to identify a new trait is fixed. So we could have 1 million images, and you would still need to spend one day, tracing outlines of bones with AI-assisted tools really quickly. And then you could retroactively measure a new trait on every image that we've ever taken. So that translates to sub-second level measurement times for new traits. So I think that's really cool, and I'd be excited for someone to do that. The other thing that I think is interesting about it is with this approach, you can measure traits that we can't really measure very easily by hand. So you could measure things like the width of the humerus at its narrowest point, which would be sort of challenging to do by hand, but it would be an easy task for a computer. And then the last thing is that we're moving into 3D. So the first image dumper or image capture setup was very bootleg. The next one is way more advanced. It's built by an engineer. It's got 5 cameras that spin around in a circle. And while they spin around, there's a platform that lifts the skeleton up and down. So you're collecting images from hundreds of different perspectives. And in keeping with the philosophy of the first setup, it only takes 2 minutes. So in 2 minutes, you can generate, this is the very first scan. It's very small. So it'll zoom in sort of frenetically in a minute. But so it only takes a couple of minutes. The image capture setup is relatively cheap, and it's extremely portable. So we can do this in any collection we want. And we can start to generate things like volumes of bones, et cetera, that we can do with a 3D scan. So I'm excited about that, even though we're just starting it now. Okay, so that's ScalaVision. That's the ScalaVision data set. Now we can get into some more interesting question-based stuff. So as I said in the beginning, I am really excited about trying to understand the interaction between climate dynamics and morphological change in birds. And for the most part, that's been restricted to the last 4 decades in North American birds for me. And this is an attempt to kind of scale that up and think about climate variability across the history of passerines, so almost 50 million years, and body plan evolution in passerines. This is work led by a postdoc at U of M named Jake Berv. He's an amazing evolutionary biologist, and I think he's a Schmidt AI and science postdoc at U of M, so he's starting to integrate AI into evolutionary methods and questions, and I think he's really at the forefront of that field, and he's asking some amazing questions, and it's really fun to work with. So I'm presenting this, but he has led all of this work in this section. We started thinking about this inspired by things like Simpson telling us that he thought most of the major diversification in life's history happened during episodes of adaptive radiation. And we can think about how you might be able to identify adaptive radiations with a phylogeny and lots of high-dimensional phenotypic data. And we can think about like Gavrelets and Losos telling us that typically there's a burst of speciation and morphological diversification soon after the beginning of an adaptive radiation rather than similar rates through time. So we have these expectations of patterns that we might see in morphological change across the phylogeny that would indicate the importance of things like adaptive radiations in the diversification history of passerines. So we set out to look at the phylogeny and see if we could see evidence of adaptive radiations across the group. And we did that with sort of three steps. So first, we tried to model the rate of body plan evolution. I'll explain exactly what I mean by that in a second, through time. But instead of just looking at this clade as a single clade, we wanted to allow the models to discover nodes at which those rates of body plan evolution changed. And then we tested whether those shifts were connected to climatic history over the period of passerine diversification. And then we wanted to take advantage of the fact that we know more about the climate and the environment now than we can say about the last 50 million years, and look at contemporary rates of body plan evolution within a current environmental context. So some of this we could do with out-of-the-box or already developed methods. So for example, this is a method developed by Clavel, essentially building a generalized multivariate Brownian motion model of high-dimensional phenotypic data on a phylogeny. So this is a model of change in this case, this example, 10 traits. And it fits the model, and in doing so, it also allows for covariance between the traits. So your evolutionary model parameterizes a variance-covariance matrix for all of these traits that you fed into the model. So what you end up with here is one species evolving through time, and each of these lines is a trait changing through time. So you can have highly covariating traits or traits that don't covary very much. And in the end, you end up with this estimate of variance and covariance. And when I say rates of body plan evolution, what I mean is the mean of the variance parameters within this variance covariance matrix. So we don't currently have the analytical tools to think about covariance yet. So really what I'm talking about when I say rates of body plan evolution, the mean of the variance parameters in this evolutionary model. So this is great. And we can apply this, it works at huge scales. So we can apply this right away to 2,000 species with 12 dimensions. But it only fits one evolutionary regime to the whole tree. It's a bit of a challenge, not really what we want to do. So we set about developing a heuristic approach to allowing the rates of evolution to change across the tree. So this is boiled down. Essentially, we generated candidate nodes at which the rate could be allowed to shift. In this case, it's any node with at least 5 species in the clade that it defines. And then we fit a baseline model, so that uniform rate model to the whole tree. And then we evaluated the improvement in model fit as we let the rate of evolution change at each of our candidate nodes. If it improved the model fit above some specified threshold, we inferred a rate shift at that node. If it didn't improve the model significantly, assume that the rate is constant through that node. So by doing that, we end up with a tree with the potential to have a constant rate across the whole tree, or the potential to have changes in diversification rates, speedups, or slowdowns at each node. So we can apply that to pasturines. And on the right here, you see a frequency distribution of rate increases in blue and rate decreases in red. So you see we identified about 250 rate decreases. So these are nodes in the tree at which we infer a decrease in the rate of body plan evolution. And then we see about 100 nodes in the tree where we inferred increases in the rate of body plan evolution. Interestingly, the magnitude of the rate increases was a lot higher than the magnitude of the rate decreases. So on this tree, we've got a few nodes where there have been big increases in body plan evolution rates, and lots of nodes where we have smaller declines in the rate of body plan evolution. And when you look at the phylogeny here, so that's the distribution of rates along the tree. So red is branches where body plan is evolving faster. Blue is where it's evolving slower. And if you just sort of squint and lean back, hopefully you'll notice a couple things. One is that at the base of the tree, things seem to be evolving faster. And that's not surprising. We see that in mammals, where you get the early origination of different orders of mammals. You see it in smaller clades like finches or anolis lizards. At the base of the tree, we tend to see these sort of bursts of morphological change. The interesting thing to me, well, I think it's interesting that we find that in pasturines. But the other interesting thing to me is that because our method doesn't treat this as one homogeneous rate, we've actually discovered that there are nested patterns of early burst evolution throughout the tree. So if you were to dig into this tree, you'd see that even as you get closer to present, you find repeated examples of fast rates at the base of the trees, followed by slower slowdowns towards. So our conclusion here is that this is really consistent with what you'd expect if there were multiple adaptive radiations that have been involved in the diversification of pasturines. I think that's pretty cool. The other thing that jumped out at us was if you look at the nodes where we see really large improvements in model fit by allowing a change in the rate of that node, and you identify them. So for example, each of these bubbles here is a node at which allowing the rate of body plan evolution to change really improved the model fit. You can compare those to taxonomy. And if you do that, there's an amazingly good correspondence between these nodes and named taxonomic groups. So for example, E is at the base of Vidua. So like you see genus and family names that correspond to nodes that we found or inferred to be places at which the rate of evolution shifts. So I think in our attempt to understand and classify bird diversity through taxonomy, we actually did kind of land on real processes that are underlying the diversification of this group. Okay, so that is certainly interesting and in line with what you might expect if adaptive radiations are important. Another component of adaptive radiations in my mind is linking to environmental change. And so here you see at the top, a reconstruction of global climate. And on the bottom, you see the results of a moving window across our phylogeny and an estimation of the frequency of rate shifts that we inferred at each point in time based on that moving window. So if you look at the black line, which is the summary of all the simulations, if the black line is high, it means that at that point in time, we inferred there to be a large number of rate shifts in the rate of body plan evolution and past rates. That line is low. We inferred it to be a period in time where there's a more consistent rate of body plan evolution through time. And we went through and we looked at the peaks of those shifts in body plan evolution rate, and they line up really well to named events in Earth's climate history. They're significant events in Earth's climate history. What does that actually mean? Well, if we can look at this a different way. So here in blue, you've got global temperatures. And in red, we've done the same sliding window approach where we moved along that global temperature and we asked what's the standard deviation of temperatures within that 5 million year period. So if you see a red line that peaks way up, that's a period of time when there's a lot of climatic variability. If you see a red line that's down close to the bottom, that's a period of time when the climate was relatively stable. And you can line that up again with our inferred frequency of rate shifts. And you find that in periods of time when the climate was really unstable or variable, we have inferred a large number of rate shifts in the rate of body plan evolution and birth. So it seems like there's some connection between climatic instability and shifts in the rate of body plan evolution in past rates. So this, I think, is cool. It's pretty hand-wavy, though. And we only have one environmental variable that we can go back 50 million years and think about. But we can take the output of our model. So we fit this model of body plan evolution rates. And at each tip in the tree, we have an estimated rate of body plan evolution for a species. It's a little bit more complicated than that, but essentially what we've done is we've said, if you take the predicted or the modeled rate of body plan evolution for each species, and you map their distributions in space, and you take the average of those rates each point in space, what does that look like? So here, if you have a red cell, it means that on average, the species in that cell are evolving their body, have a very high rate of body plan evolution. If you have a blue cell, you have lots of species that have a very low rate of body plan evolution. And we can try to understand this using lots of environmental variables. So we built a spatial model that's using things like temperature seasonality, average temperatures, precipitation, latitude, and a few other environmental variables. And we can do a pretty good job of predicting the rates of evolution across time, across space. So you see even things that are not latitudinal. So for example, at the base of the Saudi Arabian Peninsula, you see that in the actual tip rates, there's this elevated rate of body plan evolution, and we can reconstruct that in our model. So we're doing a pretty good job of understanding or predicting rates of body plan evolution across space. You can use that to sanity check some more simple models and looking at variables that we're more maybe interested in. So for example, here's a model of latitude predicting these mean rates of body plan evolution across space. This is a pretty simple spatial model that's just a quadratic model with only latitude in it. And we see this really nice latitudinal gradient in rates of body plan evolution. So species that are further away from the equator have faster rates of body plan evolution. We can also control for latitude and look at the effect of temperature seasonality, or the relationship between temperature seasonality and rates of body plan evolution across space. And we find a similar positive relationship between seasonality and rates of body plan evolution. So species that are in more seasonal environments have, on average, faster rates of body plan evolution. So why this is the case is an interesting question. But I think that, as with all of these macro scale trait-based analyses, mostly good for generating testable hypotheses. So I think we've got a couple of nice patterns that are interesting to try to understand. So just sort of stepping back and thinking about this more holistically, we've seen that the history of passerines has been characterized by nested early bursts of morphological evolution. We see that over the past 50 million years, periods of time when temperature has been hypervariable are also periods of time when we infer there to have been a lot of shifts in the rate of body plan evolution. And then if you look much closer to the present where we have richer data, we can see that there's a latitudinal gradient in rates of body plan evolution, and the rates of body plan evolution are predicted by temperature seasonality as well. Okay, so I think that's a nice example of sort of new emerging patterns that can come from large scale data sets with new types of data, like what we can build with television. I also think that by integrating some of these newer types of data that we can generate with AI with existing data sets, we can actually get toward more mechanistic understanding of long-standing spatial patterns in the world. So I'm going to talk about that here with a couple of projects that are focused on Allen's rule and Bergmann's rule. So Allen's rule is a tendency for appendages to be smaller in colder climates. Bergmann's rule is a tendency for body size to be larger in colder climates. These rules have been around for a really long time, and people are very interested in them. So there's been a lot of work on Bergmann's and Allen's rules. I am more interested in them, I'll mention in a minute, because we tend to see and interpret these temporal changes in things like size and appendage length through as temporal analogs of spatial patterns. But there are some limitations of a lot of work done looking at Bergmann's and Allen's rule. So here's a nice example with 3 lineages of barn owls, where the bill length relative to body size is well predicted by mean annual temperature. So this is a common type of analysis that you might find looking to test Allen's or Bergmann's rules. Is there a large-scale correlation between a trait and temperature? And again, these are sort of having a resurgence of interest, in part, I think, because not all species have a 40-year time series of morphological change, but there is a lot of information on spatial variation in morphology. And so if we could predict temporal responses to change using spatial gradients, that would be really powerful. And so we have seen these sort of temporal analogs of these spatial patterns. We've seen that species, at least in birds, species tend to be smaller through time, and they've also gotten longer limbed. But as I mentioned before, there's some limitations with how we've been thinking about this. So here's a thermal image of a bird, and you can see that the beak and the legs, so the unfeathered appendages in the bird, really light up. So there's a lot of heat being exchanged with the environment from the bills and the legs of this bird. So understandably, almost all of the work, and in fact, I think all of the work looking at Allen's rule in birds has focused on bill length and leg length. But there's a problem with bill length and leg length when you think about Allen's rule. The problem is that these appendages are always unfeathered. They're always exchanging heat to the environment. There's some behavioral and physiological adaptations in some species to change that, but in essence, you can't really tell if a correlation between the length of these appendages and temperature is driven by demands for heat dissipation, improved heat dissipation in hot places, or demands for cold tolerance in cold places. There are 2 possible mechanisms underlying any sort of correlation with temperature. Okay, so wings coming to the rescue. When a bird flies, lots of cool things happen. Flies, which is amazing, but it also goes crazy metabolically. So you see a massive increase in the production of heat. So chemical energy, stored chemical energy is being converted to mechanical energy. It's an inefficient process, and it generates a huge amount of heat. So here's a really nice study of lovebirds. And on the x-axis, you've got total non-evaporative heat loss from the bird. And in each of these bars are different stages of flight. So you have before it flies, while it flies, and after it flies. And the thing I want you to take away from this figure is that when a bird flies, probably not that surprisingly to you, there's a huge increase in the amount of heat that has to be dissipated. If it can't dissipate that heat, it has to stop flying because it will overheat. So how does this happen? Well, here's another set of thermal images. On the left, you see birds that are at rest. And in the left pie chart, you see their quantification of heat dissipation from different parts of the body. So you see that most of the heat that's being dissipated from these birds when they're at rest is happening through the trunk. It's happening through the chest and the back. A lot of it is happening from the head, and some of it is happening from the legs. And then when birds start to fly, you can see the brachial regions, the armpits of these birds, where there's vascularized tissue light up. And all of a sudden, you get this distribution on the right pie chart, where 86% of the heat that's dissipated by birds happens through these brachial regions of the wings. So feathers themselves are thermodynamically inert. They don't dissipate heat, but underneath the wing, there is this vascularized tissue that dissipates a huge amount of heat. In fact, most of the heat that's dissipated fly. So I think this sets up a really cool kind of natural system for thinking about Allen's rule. So wings don't seem to be important for cold tolerance. They're held against their bodies when they're at rest and a bird might be cold, and they're exposed to air and are exchanging heat with the environment when birds have jacked up their metabolism so high that they have to dissipate almost all of the heat that they're generating by flying. So there's this sort of one-way relationship between wings and thermoregulation. And so it's a nice kind of natural experiment. And with skelevision data, we have estimates of the lengths of the wing bones. So we can characterize the extent of vascularized tissue in the wings. It's an imperfect metric, of course, for how much heat is dissipated by the wing, but it's something. And we can test the hypothesis that in warmer climates, wing bones should be relatively longer because a longer wing bone and a greater extent of vascularized tissue will aid in heat dissipation. So that's our hypothesis. Wings will conform to Allen's rule, and we have some expectation that this is because of increased heat dissipation demand in warmer places, as opposed to increased cold tolerance demands in colder places. The birds are great. We can control for all sorts of stuff. So we can combine our skelevision data with AvoNet, and doing that, we can generate indices of flight maneuverability, flight efficiency. So of course, there are tons of pressures driving the evolution of wing structure in birds. And we can try to control for those. So we can control for ecology, migration strategy, latitude. And after we control for all of these things, we can look at the relationship between temperature and wing bone length in birds. And when we do that, of course, the biggest thing is allometry. So wing bone length is extremely tightly coupled to body size and overall wing length, as you might imagine. But there is a significant relationship between temperature and the relative length of wing bones in birds. And it's just what you'd imagine given Allen's rule. So bird species that are in warmer climates have longer relative wing bones than birds that are in colder climates. Okay, so that's Allen's rule. And we can also think about Bergmann's rule. Bergmann's rule is a bit shadier. I think the evidence of Bergmann's rule in birds is pretty equivocal. So you've got some people like Ashton in 2008 who didn't have a figure in their paper, so this is the best I could do. They found that it was a meta-analysis of relationships between temperature and size in birds. And they found that 76% of birds adhere to Bergmann's rule. And you can contrast that with Reimer et al., who in 2018 did a really large-scale analysis of the relationship between size and temperature within groups of birds and mammals. This is a distribution of the correlation coefficients between temperature and body size. And they found that only 14% of the species that they looked at adhere to the expectations of Bergmann's rule. 7% have a significant correlation between temperature and body size, but it's in the opposite direction from what you'd expect. And the vast majority of them have no significant relationship. So it's really, I think, not clear that birds follow Bergmann's rule. That's some sort of phenomenological, empirical evidence for that. But we also have ecophysiologists kind of screaming from the rooftops that Bergmann's rule doesn't make sense. So going back to the 50s, you have Scholander arguing that Bergmann's initial explanation for his rule, which was that as individuals get bigger, the ratio of surface area to volume gets smaller, and so cold tolerance should improve. So Scholander was saying, this is ridiculous. If you're an endotherm and you want to reduce your heat loss, you would not make your body bigger. You would do something much easier. There's so many levers that you can pull. You can just alter your insulation a little bit, You can change your vascular constriction a tiny bit, and those small changes will have a really, really big impact on energy budgets and heat dissipation. Whereas you'd have to do a massive change in body size to achieve the same thing. So for a long time, people have said that about spatial patterns. We're also seeing people say that about temporal patterns, mostly Andreas Nord and co. And they have done amazing stuff where they've taken long-term trends in body size and appendage length, and they've modeled the impacts of those changes on the thermodynamics of birds, and essentially shown that the changes in heat dissipation dynamics are more or less negligible as you change the body sizes and appendage lengths to the extent that we observe that they have changed. They've also done some new experimental work showing this. So I think the ecophysiologists are kind of saying, like, this doesn't really add up. So that's all to say, it seems worth testing within our data set. So this is work led by Charlotte Probst, who's a PhD student in Michigan. And we set out to ask whether or not bird skeletal size adheres to Bergmann's rule. And then in honor of the ecophysiologists, we tried to develop a more thermodynamically informed metric of heat dissipation, demand, and cold tolerance. And we thought that relative mass would do that. So this is the amount of mass in a species of bird given its skeletal size. So a high relative mass would be a bird that's heavier than you would expect given its skeleton. And I'll talk about why that's significant. So we can do that by combining our skelevision data with existing data on bird masses. And so we do that with skeletal size. So this is these are coefficients from a comparative phylogenetic model trying to predict skeletal size of a couple thousand species of passerines using the maximum and minimum annual temperatures in their ranges, precipitation, and an estimate of or a proxy for ecosystem productivity and range size. And on the left here, you have among species. So these are looking at 2,000 different species. And there's no relationship between any of these environmental variables and variation in structural size. Almost all of the variation is explained by phylogeny. When you look at skeletal size within species, so we have a few species where we have enough data to actually look within the species, you see a very similar picture. There's sort of a marginally non-significant relationship with temperature in the way that you'd predict with Bergmann's rule, but it's marginally non-significant, and more importantly, it explains almost no variation in body size. So there's really no evidence of Bergmann's rule in the skeletal size of passerines. But when you look at relative mass, it's a very different story. So here on the left again, you've got the model output looking across species, so looking at species, and you see that the temperature variables become important in the way that you'd expect given cold tolerance and heat dissipation demands. So basically, in warmer places, you have birds that have lower mass than expected given their skeletal size. And the mass relative to skeletal size is often used as an index of insulation. So the assumption is that increased mass is either because of increases in fat or increases in muscle. And both of those increases alter heat exchange with the environment dynamics in very similar ways. When you look within species, this pattern disappears. So really what we're finding is no evidence of Bergmann's rule in skeletal size, and pretty strong evidence that temperature does predict things like insulation across birds. I should add that these models of relative mass, the fixed effects explain a lot of the variation in relative mass. It's pretty strikingly different from the models of skeletal size. Okay, so as much as possible with these large-scale data sets, I think that the addition of a new type of data, so a different anatomical system, into our existing understanding of bird morphology has let us kind of tease apart a little bit the mechanisms underlying large-scale patterns. So again, I think passerine wings, sort of amazing. It seems like passerine wings and the evolution of passerine wings has been at least in part shaped by thermoregulatory demands, which I think is very surprising, at least it was to me. And I think it's a nice example of Allen's rule. So these wing bones conform to Allen's rule, but given that they are not that important to cold tolerance, it seems to be an example of Allen's rule, probably driven by increased heat dissipation demand in warmer places. And then the Bergmann's rule stuff, I think, shows you that this sort of more nuanced understanding of heat exchange with the environment leads to different conclusions about large-scale patterns in morphology as they relate to size and temperature, essentially showing that skeletal size doesn't conform to Bergman's rule in passerines, but this more sort of ecophysiologically informed metric of heat dissipation capacity does. So just stepping back again, I hope that people are excited about ScalaVision. The data are about to be publicly available, but if anybody's interested in them, let me know. We're more than happy to share them, have at it. That project is growing and expanding. And it's interesting for me in that it's not very question-based, the expansion. And so if you have groups that you're interested in, you can prioritize those for us. So I think that's a fun example of how AI can kind of help us generate new types of data at really large scales and really quickly. And I think that that's exciting. I'm happy to talk with people about that. I'm really excited about the relationships between climate and climate variability and rates of body plan evolution in passerines. I think it's really sort of thought-provoking to see these new gradients and things like latitudinal gradients and things like rates of body plan evolution, and thinking how they fit together with the latitudinal gradient species diversity, for example. And then, so I think that these new types of data can reveal new patterns to us. And then finally, I thought it was really satisfying to kind of combine this new type of data with existing data and our understanding of the physiology of birds to kind of get a better mechanistic understanding of these longstanding patterns that we've observed in nature. So again, I want to thank you for the invitation to talk today. I'm thrilled to be finally at the Field Museum. Hopefully it'll be the first of many longer visits. If anyone has questions or are interested in anything, please feel free to shoot me an email, and I'd be happy to take any questions.
All right. It's on. Good. I have the microphone. We've got any questions? You guys are going to ask the same question?
Great talk, Brian. Lots to think about. I'm curious if you tried the same analyses with that you did with body plan evolution, if you just plugged in the AVANET traits instead, if you'd get the same results, and if not, what would be different?
Yeah,
and which traits would underline it?
That's a great question, and I don't know the answer to that. My guess is that the AVANET traits are measuring similar aspects of the phenotype. It's missing some of the traits that we have. So like we have keel length, for example. So I think that you would probably get, you could definitely do that. And I think that you might get broadly similar results, but I think you'd have sort of less, you'd have less understanding of kind of the modularity of the evolution. So one of the things that I haven't shown is, you don't give these models any information. Like we don't tell them anything about taxonomy or climate or anything. It's just traits. And you can pull out lots of amazing things. Like you can pull out the variance covariance matrix, and you can see within that variance covariance matrix what is evolving together. So you'll see that like the model discovers that elements of the wings evolve together. They covary together way more than elements of the wing and the leg bones, for example. And so with the AVANET data, you lose all of that because like the AVANET data has like one metric, one measure of a wing of a leg bone. It's got one aspect of well, two aspects of sort of wing plumage. And so I think you would maybe recover the same overarching pattern, but you would lose a huge amount of information that has to do with like the high dimensionality of the data. It's just sort of higher dimensions. Yeah, it's a great question. We should totally run this with the AVANET data and see what happens.
Hey, Brian. So one of the reasons this data set from the Field Museum is so important is because it was measured by one person, Dave Willard, for the last 40 years, and that has a lot of power in it. So how much power does your ScalaVision have? I mean, have you measured them more than once? What's the rate of variation? And what happens if a bone is slightly torqued or twisted a bit, which we all know that we see that in skeletons?
Yeah, that's a wonderful question. And ScalaVision is no Dave Willard. It is, so we have done a few things to try to figure this out. So we have we've had one person measure a bunch of the elements from a bunch of the skeletons. And then we have compared the skelevision estimates of that same specimen to the handmade measurements. And then I think more to the point of your question, we've also taken the same skeleton and we've dumped it out and photographed it and then put it back and dumped it out and photographed it again. And we've done that 30 times for a bunch of skeletons. And then we've compared the variation in the trait estimates. So it's measuring the same femur, for example, 30 times with 30 different orientations of the bones. Sometimes they're overlapping other bones so that they're lifted off the surface a little bit. And we can quantify the precision that way. So essentially what we found is that there is some error. The error is usually less than a millimeter. And that's true both for the accuracy and the precision. It's not uniform across elements though. So some of the elements are harder for the model to identify and measure than others. And so there is some variation in the error rate based on the element that you're talking about. So it's definitely not the same as having the same person measure a specimen, all the specimens. But if you compare it to the AVONet data, for example, and you look at the error, so AVONet did something similar. It had people measure the tarsus it had lots of people measure the tarsus for the same species, and then looked at the variation among those measurements. And I think they also had people measure the same specimen and looked at the inter-measure error. And those error rates are comparable to the television error rates.
What happens if you were to take the information and look at the shape of the beak, for example, quantitatively and mathematically? Is there how does it do in that perspective?
Yeah.
Not just linear measurements, but when you're using them to measure the shape of something.
Yeah, that's a great question. And I think is a priority for what we should try to do next with all these images, but we haven't looked at that at all. And so that's sort of, I think what I was trying to get at with it's not just that you can do measurements that we can do by hand, but faster. We can get things like shape, quantifications of shape, can't really do by hand. And one of the exciting things about it is, we've already trained the model to identify the beak, for example.
Right.
And so it wouldn't actually be that much work to get it to measure something different about the element because it's already found the element. One thing that I didn't talk about is that it's not perfect. So, and actually, so one of the things that's interesting about it is it's not perfect. And we actually have, for some of these elements, we have like 50% missing data. So you'll run a specimen, you'll take an image, and maybe for whatever reason, you can't see either of the tarsi. And so it doesn't have, it knows that it doesn't see them, and it doesn't give you an answer. It says, I don't have an answer. But then you can actually infer the lengths of those bones because the data are so highly dimensional. Like if you know 12 dimensions of a bird skeleton and you know 11, you can actually infer the missing data really well. And so we've done a lot of work to try to figure out how accurate are these inferences, and they're insanely accurate. So with a data set that's this big and this rich phenotypically, you can draw information from all the different elements to estimate the lengths or shapes of missing elements.
Well,
I know
that's
you didn't ask
amazing.
that, but I think it's cool.
And what about things like hyoid apparatus, like bones that are super thin and
It should be able to do that. So the resolution is, especially on the new setup, the resolution is amazing. It's well below a millimeter, and so it should be able to do really fine bones. And computers are a lot better than us at some things, like looking for a variation in color. So it should be able to do a really good job of these really fine parts.
and it's really cool. I'm so happy our specimens are used in this.
Thanks, that was really cool. I was wondering about your results for relative body size. And if I was reading the figures correctly, you recovered a signal for year in the within species data that was very similar to the temperature signal in the across species data. And I'm wondering if that's a signal of, I don't know, a warming planet
Yeah.
showing in relative body size.
Yeah, it's a great question. And we don't know, well, within these data, so the way that within species analyses are done, the samples are not well suited to look for changes through time. So we haven't selected individuals in order to look at changes through time. It's mostly in there to control for anything that might be happening. And so we haven't put much stock into that year effect because we just don't think that this is like a good setup for looking at temporal trends. But we've looked to see if the variables are correlated, and they're not correlated. So I'm not sure what's driving that result. It's also, you may have noticed, it's like in the opposite direction from what you'd expect, kind of, at least within the skeletal side of things. So I don't know what to say about that, except that it's not, some issue with multi-collinearity.
Got time for one more.
We're gonna have a lot of 3D stuff, right? What do you expect to learn from the 3D reconstructions? Is it just more precision in the measurements, or is it like something fundamentally different that we can get from 3D data?
Well, I think we've been talking a lot about the potential to use 3D data for things like if you have fragmentary remains of birds, got a bag of 10,000 little bones, and you want to identify, like classify them to species. I think that would be really hard to do with two-dimensional data, but with three-dimensional data, especially like the resolution on this is extremely high, and so you will be able to kind of capture subtle variation in 3D shapes. That is the type of variation that paleontologists use to identify specimens or identify fossil remains. So that's one of the first things I think we're going to try to do with it is train a species classifier basically, and see if we can kind of speed up the classification of lots of fragmentary parts of bones. But yeah, I don't know. There's lots of other stuff to do. Like you can think about like the scaling of bones and whether or not that varies with life history traits like migration. And you can. Well, I think there's lots of interesting stuff, but that's our first goal is classifier.
All right, we're coming up on the hour, so I'm going to go ahead and stop that now. If you want to come up and ask Brian some questions, please do. Thank you all for coming, and we really appreciate it. Thanks, Brian.